3D bioprinting, also just known as bioprinting, is a relatively new technology that, in theory, would allow humans to fabricate nearly any tissue or organ from scratch. The fundamental idea behind bioprinting is quite similar to that of ordinary 3D printing, in which a material, usually plastic, is printed, one layer at a time. Instead of plastic, however, bioprinters use bioink, usually composed of cells suspended in a special gel known as a hydrogel, which helps to protect, nourish, and hold the cells together. Some bioinks use a single type of cells, while others contain multiple types of cells, or multiple bioinks are used side by side, each with a different cell type. There are 3 categories of bioprinting that I will be discussing in this video, which differentiate in the method by which they turn the bioink into a specific shape. Note that due to the fact that this is an emerging technology, the exact name and classification of various methods vary from source to source. The concepts, however, are universal. I choose to group the various bioprinting methods into the following categories: extrusion-based bioprinting, droplet-based bioprinting, and energy-based bioprinting. Extrusion-based bioprinting is similar to what most people would think of when they think of conventional 3D printers. It involves forcing continuous filaments of a material through a nozzle in a controlled manner to create a 3D structure. The material is bioink, which, as I said before, usually consists of cells and a hydrogel. The filaments are forced through a nozzle by either pneumatic pressure, which is basically air pressure, or mechanically derived pressure, which comes from things like pistons or screws. The bioink must be stabilized quickly, or else it will not retain its shape. The bioink can be stabilized in a number of ways, largely depending on the hydrogel that is being used. As an example, during printing, the bioink can be stabilized by spraying a mist with a cross-linking agent dissolved in it. By the way, cross-linking just means linking one polymer chain to another, and this is done to stabilize bioink. In contrast, droplet-based bioprinting deposits discrete volumes or droplets of bioink onto a surface. Droplet-based bioprinting methods include Inkjet-Based Bioprinting, Microvalve-Based Bioprinting, and Laser-Induced Forward Transfer Bioprinting. As with Extrusion-Based Bioprinting, the bioink must be quickly stabilized in order for the structure to retain its shape, and the exact manner in which this is accomplished depends on the hydrogel being used. Inkjet-based bioprinting shares a lot in common with traditional inkjet printing. Here's how it works. At a high level, a pulse of pressure is used to eject a droplet of bioink. The pulse of pressure can be generated in one of 2 ways, using thermal mechanisms or piezoelectric mechanisms. In the thermal mechanism, a small surface of the bioink is heated and vaporized to create a bubble, which occupies a larger space than the liquid bioink did, creating pressure, forcing a droplet of bioink out of the nozzle. Once the bubble collapses, a bit of bioink is sucked from the reservoir, refilling the chamber, and the process is repeated. In the piezoelectric mechanism, an electric current is applied to a piezoelectric actuator, causing the chamber to deform slightly, forcing a droplet of bioink out of the nozzle. By the way, a piezoelectric actuator is a device that responds to an electric current by stretching and bending. Then, when the electric current to the piezoelectric actuator ceases, the shape returns to normal, and a bit of bioink is sucked from the reservoir, refilling the chamber, and the process is repeated. Next, microvalve-based bioprinting involves small valves that can be accurately opened and closed with electromagnets to deposit droplets of bioink, which is under pressure, usually pneumatic pressure, meaning air pressure. Laser-induced forward transfer bioprinting uses lasers to accurately position cells on a substrate, or the place where the tissue will lie. Laser-induced forward transfer bioprinting consists of a laser, a focus lens, a ribbon, and a substrate. The ribbon could contain a sheet of transparent quartz glass with a very thin gold coating and a coating of bioink. When the laser reaches the gold, it heats it and greatly expands it, propelling a very small amount of bioink to the substrate, which will have been coated with hydrogel to dampen the kinetic energy of the droplet of bioink. This laser is quite precise, and hence this method is also quite precise. Finally, in energy-based bioprinting, a focused energy source, often a laser, is used to selectively solidify or stabilize a bioink. This method differs from extrusion-based bioprinting and droplet-based bioprinting in that the bioink is already in place. Perhaps the most notable method of energy-based bioprinting is stereolithography. In stereolithography, a laser is employed to selectively harden a small amount of bioink, which contains a light-sensitive hydrogel. This substance lies on a platform that is then moved away from the laser by a small amount. If, in doing so, the platform is immersed into bioink, then a fresh coat of bioink will flow on top of the now hardened layer of bioink, or if the platform has sidewalls, then a fresh layer of bioink can be coated separately. This process is repeated, eventually leaving you with a solid 3D structure once the liquid bioink is washed away. Each category of bioprinting has its own pros and cons. I won't bore you with all the specifics. However, as an example, laser-induced forward transfer bioprinting is precise and has a high printing resolution. Nevertheless, it is expensive, cumbersome, and time-consuming. Hence, different methods are used for different needs. Interestingly, there are approaches being developed that combine different bioprinting methods in order to maximize efficiency. Maximizing efficiency is crucial for bioprinting certain structures like organs. In general, organs must be printed quite quickly, and yet they have certain parts that contain lots of details that consequently require bioprinting with a high resolution. Other parts don't need to be printed with such precision, and time can be saved by not printing at such a high resolution. So, by combining certain methods that print slowly with a high resolution with those that print quickly with a lower resolution, one can optimize the bioprinting process. While these bioprinting methods are based on 3D printing, living things develop and change over time. Hence, bioprinting can often be thought of as 4D bioprinting, where the cells in the printed tissue proliferate, interact, and change in various ways over time. In fact, certain chemicals are often added to the bioink to influence the behavior and development of cells. Also, over time, hydrogel is meant to slowly degrade and be replaced by the native extracellular matrix. The extracellular matrix is the non-living material that cells secrete, which fills the spaces between cells, protects cells, and holds cells together. Sounds familiar? That's because hydrogels are meant to resemble the extracellular matrix. Now that you understand how the various methods of bioprinting work, I would like to highlight some cool applications. Perhaps most notable is the ability to print entire fully functional organs from scratch. Doing this poses unique challenges due to the intricacy of organs, but even printing complex organs will likely be viable in the near future. If this is the case, the demand for organs on organ transplant lists can finally be equal to the supply of healthy organs. With bioprinting, healthy cells can be taken from a patient's organ and be allowed to proliferate a little bit, if possible, and can then be used in the bioink. By taking cells from a patient rather than from some other individual, the probability of an organ being rejected by the patient's immune system is drastically reduced. In this way, bioprinted organs can be superior to foreign donor-derived organs, And if creating custom organs from a patient's own cells is not a viable option for some reason, because of urgency in implanting a new organ, for instance, there is still the option of having generic, pre-made organs. Also, a special type of cells called stem cells introduces some pretty cool possibilities, allowing for personalized organs, even if directly acquiring the necessary cells is impossible. Stem cells are basically cells with the ability to change their functionality based on their environment. All of the cells in a human's body with specific functions actually originate from certain types of stem cells. So, instead of being forced to take certain cells from a patient, like healthy cells from a failing organ, which may be very hard or even impossible to acquire, cells can be taken, say, from the patient's skin, and in a lab, they can be artificially transformed into stem cells, which can then be induced into developing into the different cell types that are needed to bioprint a healthy new organ. As previously mentioned, stem cells allow for increased flexibility in bioprinting. However, note that actually working with stem cells is quite challenging, and even bioprinting simple tissues with them at this time is very difficult. Bioprinting tissues is simpler than printing entire organs, and is still extremely valuable in a number of ways. For example, bioprinting can be used to create vegan meat or leather. To do this, a small number of certain cells must be collected from, say, a cow. Then, by carefully controlling the conditions in which the cells are held, these cells can proliferate. After this, the cells can be collected and bioprinted in a strategic manner, and in a bit of time, what remains is meat or leather. Besides moral considerations, with investment and development in this field, cultivating meat and leather in this way could be much more efficient and have a much smaller negative effect on the environment in terms of things like carbon emissions, land use, and freshwater use. Also, bioprinting could radically change the process of developing and testing new drugs. Today, pharmaceutical companies test the efficacy and safety of a potential new drug on animals. If a drug proves to have the desired effect on animals, primarily mice, without harsh side effects, then the drug is tested on humans. Often, there is no desired effect on humans, rendering the money invested in developing that drug a waste. In fact, 90% of drugs that showed promise in animals have no significant effects on humans. Occasionally, the drugs can even harm the humans being tested. This testing process is extremely expensive and inconvenient. With bioprinting, human tissue or even organs can be bioprinted that more accurately mimic the human condition and thus are more suitable for testing drugs. An organ on a chip is a simplified, miniature version of an organ that aims to mimic a real organ so that it can be used to test a new drug. Technically, organs on chips are not inherently related to bioprinting, but bioprinted tissue can be used to enhance such devices with better positioning of cells and reproducibility. In the near future, it may be possible to have a nearly perfectly accurate system for testing drugs by linking organs on chips together in a microfluidic circuit, which would be known as a human on a chip. What this means is that to test an oral drug, for example, the drug could first be introduced to a partially bioprinted gut on a chip, which would absorb the compound and send it to a partially bioprinted liver on a chip, where it is metabolized. The metabolites are then sent to other partially bioprinted organs on chips, including the kidney on a chip, heart on a chip, lung on a chip, and bone on a chip. Throughout this process, the organs are tested for any intended or unintended response to the drugs. A similar pipeline could be used to test the safety and effectiveness of other forms of drugs. Note that in this process, organs on chips need not have the full functionality of particular organs. Instead, they only mimic the parts of the organ that are valuable in testing the effects of a new drug. Also, instead of introducing a disease to healthy animals and then testing various drugs to treat the animals, a disease could be directly introduced to a human on a chip, reducing the amount of unnecessary suffering. In summary, using bioprinting to test new drugs could be both more efficient and more ethical. In a similar way, the safety of new cosmetics could be tested without using animals, but using bioprinted tissue instead. Although it would come with important challenges of its own, bioprinting plants is a potential future application of bioprinting, with the possibility of creating new plant varieties with desired traits for agricultural use. Looking ahead, if or even when bioprinting fully functional organs is common practice, the following questions naturally arise. Can bioprinting be used to create organs with abilities superior to the organs that we are born with? For example, can we bioprint eyes with supervision, or bioprint super efficient lungs, as found in elite athletes? Is such a use of bioprinting ethical? Will bioprinted organs and superorgans be reserved for the rich? These are important questions that we must ask ourselves as this technology is further developed. Thanks for watching.